Good morning. Myself, Dr. Ragasudha, Head and Assistant Professor, Department of Microbiology, Government City College, Hyderabad, is here to discuss on antibodies or also known as immunoglobulins. Antibodies are glycoproteins which are naturally produced in response to the invading foreign particles or antigens. Immunoglobulins function as antibodies, the antigen binding proteins that are present on the B cell membranes and also are secreted by plasma cells. Membrane bound antibody confers antigenic specificity on B cells. Antigen specific proliferation of B cell clones is elicited by the interaction of membrane antibody with antigen. Secreted antibodies circulate in the blood. They serve as the effectors of humoral immunity by searching out and neutralizing antigens or marking them for elimination. All immunoglobulins share certain structural features, bind to antigen and participate in a limited number of effector functions. Immunoglobulins are attached to the B cell membrane while antibodies float in the circulation. The main difference is that immunoglobulins has transmembrane domain in order to be attached to the plasma membrane, while the transmembrane domain is absent in antibodies. Thus, proteins that recognize and bind to a particular antigen with very high specificity are known as antibodies. Antibodies are made in response to the exposure to the antigen. One virus or microbe may have several antigenic determinant sites to which different antibodies may bind. Each antibody has at least two identical sites that bind antigen called antigen binding sites. Valency of an antibody, that is the number of antigen binding sites, is mostly bivalent. The serum antibodies produced in response to a particular antigen are heterogeneous because most antigens are complex and contain many different antigenic determinants. The immune system usually responds by producing antibodies to several of them. This response requires the recruitment of several clones of B cells. Their outputs are monoclonal antibodies, each of which specifically bind to a single antigenic determinant. Together, these monoclonal antibodies make up the polyclonal and heterogeneous serum antibody response to a immunization antigen. They belong to a group of serum proteins called immunoglobulins. Now, let us see the discoveries behind the structure of immunoglobulins. It has been known since the turn of the century that antibodies, the effector molecules of humoral immunity, reside in the serum. The first evidence that antibodies were contained in a particular serum protein fractions came from a classical experiment by Tiselius and Kabat in 1939. They immunized rabbits with the protein ovalbumin and then divided the immunized rabbit serum into two aliquots. Electrophoresis of one serum aliquot revealed 4 peaks corresponding to albumin and alpha, beta and gamma globulins. The other serum aliquot was reacted with the ovalbumin and the precipitate that formed was removed. The remaining serum protein which did not react with the antigen was then electrophorized. A comparison of the electrophoretic profiles of these two serum aliquots revealed that there was a significant drop in the gamma globulin peak in the second aliquot that has been reacted with the antigen. Thus, the gamma globulin fraction was identified as containing serum antibodies which are called as immunoglobulins to distinguish them from any other proteins that might be contained in the gamma globulin fraction. Here in the picture we can see the two graphs produced of the two aliquots. The first graph containing 4 peaks of albumin, alpha, beta and gamma globulin, and the second graph represents the second aliquot which was treated with the antigen, where we can see a significant drop in the graph of gamma globulins. Moving on to the next discovery, which deduced the antibody structure. Our knowledge of basic antibody structure comes from a variety of experimental observations. In 1950s and 60s, experiments by Rodney Potter and by Gerald Eldman elucidated the basic structure of the immunoglobulin molecule. Eldman's and Potter's experimental approaches were quite different. Potter cleaved the immunoglobulin molecule with an enzyme to obtain fragments, whereas Eldman dissociated the molecules by reducing the interchain disulfide bonds. The result attained by these two approaches complemented each other. Potter and Eldman first separated gamma globulin fraction of serum into high molecular weight and low molecular fractions. They both studied the low molecular weight fraction, which contained 1,50,000 molecular weight gamma globulin designated as immunoglobulin G. Potter subjected IgG to brief digestion with the enzyme papain and separated the fragments. Although papain has general non-specific proteolytic activity and will eventually digest the entire IgG molecule, brief treatment cleaves only the most susceptible bonds. Papain digested IgG produced 2 identical fragments, each with a molecular weight of 45,000, called FAB fragments, or also known as fragment antigen binding, because they have antigen binding activity, and one fragment with a molecular weight of 50,000 called FC fragment or fragment crystallizable, because it is found to crystallize during cold storage. A similar experimental approach but with the enzyme pepsin was tried by Alfred Nisonoff. Brief pepsin digestion generated a single 1,000 molecular weight, 1 lakh molecular weight fragment composed of 2 FAB like fragments and designated as FAB2. Unlike FAB fragment, the FAB2 fragment was able to precipitate antigens. However, the FC fragment was not recovered from pepsin digestion because it has been digested into multiple fragments. The multi-chain structure of IgG was first suggested by the experiments of Eldman and his colleagues and later confirmed by Potter. Potter subjected IgG to mercaptoethanol reduction and alkylation, a chemical treatment that irreversibly cleaves disulfide bonds. The sample was then chromatographed on a column that separates the molecules by size. This experiment reveals that 1,50,000 molecular weight IgG molecule was composed of 2 50,000 molecular weight polypeptide chains designated as heavy chain and 2 25,000 molecular weight chain designated as light chain. The puzzle was to determine how the enzyme digestion produced FAB, FAB2 and FC was related to the heavy chain and light chain reduction products. Potter answered this question by using antisera from goats that has been immunized with either FAB fragments or FC fragment of the rabbit IgG. He found that antibody to FAB fragment could react with both H and L chains, whereas antibodies to the FC fragment react only to the H chain. This observation led to the conclusion that FAB consists of portions of heavy and light chain and FC contains only heavy chain components. These results, Potter and Eldman proposed a structure for IgG. According to this model, IgG molecule consists of 2 identical H chains and two identical L chains which are linked by disulfide bridges. The enzyme papain cleaves just above the interchain disulfide bonds linking the heavy chain, whereas the enzyme pepsin cleaves just below the bond, so that the two proteolytic enzymes generate different digestion products. Mercaptoethanol reduction and alkylation allow separation of the individual heavy and light chains. Here in the picture we can see when IgG is digested with pepsin, how it produces Fc fragments and FAB2 fragments. When it is digested with papain, it produces 2 FABs and 1 Fc fragment, and with mercaptoethanol reduction, it produces individual heavy and light chains. Antibody structure. Antibody structure, in monomer, it is a flexible Y-shaped molecule with 4 protein chains. It comprises of 2 identical light chains and 2 identical heavy chains. Variable region, 2 sections at the end of the Y arms have variable regions, contains antigen binding sites called FAB, identical on the same antibody but vary from one antibody to the another antibody. The constant region, it is the stem of the monomer and lower part of the Y arm. Fc region is the stem of the monomer only, important because they can bind to the complement or cells. Let us get into the details of the structure of the immunoglobulin. All immunoglobulin molecules consist of 2 identical light chains and 2 identical heavy chains held together as a tetramer by interchain disulfide bonds. The arrangement of 4 polypeptide chains in an immunoglobulin molecule gives it a Y shape. There are 2 types of light chains known by the Greek letters kappa and lambda. An immunoglobulin has either K or lambda chain, kappa or lambda chain, but not both. There are 5 types of heavy chains designated as gamma, alpha, mu, epsilon and delta. Depending upon the type of the heavy chain, 5 different types of immunoglobulins are identified as IgG containing gamma globulins, IgA containing mu, IgM containing rho, IgE containing delta, IgE containing epsilon and IgD containing delta. Each type of heavy chain can combine with each type of light chain, but within the same antibody molecule, the 2 heavy and light chains are of the same type. Variable region. The half of the light chain towards N terminal is called variable region, or termed as or designated as VL, variable light chain, and the other half towards C terminal end is called as constant region. Similarly, 1/4 of the heavy chain at the N terminal end is called variable region, and 1/3 at the C terminal is called constant region. Domains or regions. Each chain can be divided into domains or regions. In each of the 2 light chains, there are 2 interchain disulfide bonds giving rise to 2 domains, one each in variable and constant region. Similarly, there are 4 intra-chain disulfide bonds in the heavy chain giving rise to 4 domains in each of the heavy chain. One domain appears in each variable of the heavy chain and 3 in the constant region. Papain digestion. When immunoglobulins are subjected to papain digestion, 3 fragments are produced. 2 of them retain the ability to bind with the antigen, hence they are called as antibinding fragments. The third one does but is easily crystallizable and hence called Fc fragment or crystallization fragment. The point of the papain cleavage is called hinge region. What is this hinge region? It is highly flexible amino acid stretch present between CH1 and CH2 regions of the heavy chain. Hinge region is absent in IgM and IgE. They possess high hinges like IgM and IgE possess hinges like domains with approximately of 110 amino acids. Hinge region connects two heavy chains with many disulfide bonds. The number of bonds differ in different immunoglobulin isotopes. Hinge region is rich in cysteine and proline and is flexible. The flexibility gives IgG, IgD and IgA segmental flexibility. As a result, the two FAB arms can assume various angles at each other when antigen is bound. This flexibility of the hinge region can be visualized in electron micrograph of antigen antibody complexes. This is a picture representing the structure of immunoglobulins with showing variable regions and the constant regions, N terminals and C terminal. N terminal is the terminal where antigen binds, C terminal is the tail part of the immunoglobulin, and the hinge region is the region where the upper part binds with the C terminal, or the N terminal binds with the C terminal. Variable region is represented in the red color with antigen binding sites, whereas the constant region is in blue color. Heavy chain consists of 3 constant regions and 1 variable region, whereas a light chain consists of 1 variable region and 1 constant region. Antibody structure. Here we can see the enlarged antigen binding site. The epitope of the antigenic determinant. The epitope present on the antigen binds with the antigen binding site in this fashion at the variable region. Thank you. The basic structure of an antibody molecule consists of 2 identical light chains and 2 identical heavy chains, which are linked by disulfide bonds. The amino terminal 110 amino acids in each heavy and light chain consists of a variable VH and VL domains. The remainder of each light chain consists of a single constant domain. The remainder of each heavy chain contains 3 or 4 constant domains. In any given antibody molecule, the constant region contains one of the 5 basic heavy chain sequences, mu, delta, alpha, gamma and epsilon, and one of the 2 basic light chain sequence consists of either kappa or lambda type. These sequences are called isotopes. The heavy chain isotype determines 5 classes of antibodies, namely IgM, IgG, IgD, IgA and IgE, and the effector function of the molecules. Each of the domain in the immunoglobulin has a characteristic tertiary structure called immunoglobulin fold. The Ig fold structure is also present in a large group of membrane proteins. Within the amino terminal variable domain of each heavy and light chain, there are three hypervariable regions also called as complementarity determining regions. They are formed by some of the loops that connect the beta strands of these domains. The 3D conformation of an antibody molecule, these regions are brought into proximity, forming its antigen binding site and determining its specificity. Immunoglobulins are expressed in two forms: secreted antibody produced by plasma cells and the membrane bound antibody present on the surface of the B cells. Membrane bound immunoglobulins associate with a heterodimer called Ig alpha Ig beta to form B cell receptors. The antigen specificity of a B cell is determined by the membrane bound antibody that it expresses. Ig alpha Ig beta heterodimer of the B cell receptor mediates the intracellular signals that lead to the B cell activation following interaction with the antigen. Here we conclude with the structure of the immunoglobulin or antibodies. Thank you.